We are so thankful to have Davida Smyth with us today. Davida is an associate professor of molecular microbiology at Texas A&M in San Antonio, but she did come to San Antonio from the New School in New York City. At the New School, she was, and also in San Antonio, she's been identifying and analyzing SARS-CoV-2 from wastewater. So I think Davida mentioned that she's going to be analyzing some this afternoon after this talk. She conducts research with her undergraduate students, and she wears many hats. So today she's going to be talking to us about her work with SENCER, and the title of her talk is STEM Educators as Civic Educators. And welcome, Davida. We're so thankful that you're here.
Oh, no problem. My pleasure. I feel very honored to be invited to such an illustrious group. All right, let me just share my screen real quick. There we go. Oh, that's not what I meant to do. Slideshow. Now, there we go. All right. So Melanie, as you all know, I adore her. So when she asked me to do this, I was absolutely delighted. And of course, I'd be here. And I'm going to talk to you all about a little bit about how STEM educators can be civic educators. And we started you all in the chat there, thinking about how you all consider that prompt. Okay. But I'm going to send you off into breakout rooms right away. So you have to work for this talk. And I'll give you like 5 minutes, roughly 5 minutes, just to think about what issues you care about. And then in parallel with that, I want you to think about what issues your students care about. All right. So you might be thinking about AMR, but your students probably think about parking. Okay. So what are the issues that you think about, and what are the issues that your students think about? 5 minutes off into breakout rooms, and I'll bring you back. Got it. Folks are cool. All right, you're all coming back. All right, so I'm sure you all came up with loads of ideas, and so feel free to start putting in what you care about and what your students care about. And what I wanted you to consider in that prompt, it seems obvious, but that oftentimes what we care about is not what our students care about, and that's really important. It was one of the first things that kind of blew my mind when I actually asked the students, which we often don't do. We just launch into, we're going to study this issue today. We don't often ask our students, what are you freaking out about right now? And that's something we should always consider starting with. All right. So SENCER, just to let you know a little bit about SENCER. SENCER is the organization that changed my life. Not kidding, not joking there. It changed my life. And it made me allowed to understand that, it helped me understand, I should say, that you can build science understanding by including civic issues in your classes, right? And build civic capacity, which is really important for our students, because more often than not, they say to you, why do I need to do this? Why am I in this class? I don't like, what's biology for? Blah, blah, blah. How many of times have we heard this, right? But it also allows students to consider science as part of the whole, of the greater picture, right? That, you know, science relates to history, it relates to our cultures, our economies, legal issues, ethical factors. And we're thinking that way now, but pre-COVID, I mean, a lot of us wouldn't have even considered these kinds of things in our classes. And it also helps students kind of understand political structures, civic governmental structures that will impact have an impact on science, and that science itself can impact as well, right? So that's really important. So how does SENCER begin? I have QR codes here, so you can get these slides. You'll be able to boop them and bring them home with you, right? But SENCER started with Monica Devanis, who is also one of my favorite humans, right? She started a course back in the day looking at HIV and AIDS, when HIV and AIDS were effectively becoming a huge problem at the time, right? So she rebuilt a whole course through HIV and AIDS, a molecular biology course at the time, which was cool. And she talked about the economics of it, the legal aspects of it, the social context of it, and made a course, the Biomedical Issues of HIV-AIDS. And I apologize, it's blurry, but this is the only image I have of that. But in essence, so basically people take courses and they can basically take these kind of disciplinary context courses and reframe them through issues, right? So molecular biology, which many of us teach, becomes a course on cancer. General chemistry, which is really hard to redesign, becomes exposure to toxic chemicals. You can have environmental courses that are focused on Southwest Florida, pollinators, sustainability, and so on and so forth. So everything can be reframed when you go at it from a SENCER point of view, okay? And this picture, for some reason, always takes its time, but these are all, we 55 peer-reviewed model courses. So there's something for everybody in there. And we're actually working on, I'm recruiting people right now to help me kind of go in and look at these courses and kind of bring them into the present day. But we've loads and loads of examples with assessments and activities and everything else that were contextualized at their time to what was going on as well. So that's something to consider. And if we think about SENCER, the resources that we have these model courses, but we also have pearls of practice, like little short kind of things that you can do. We have backgrounders on topics that are sometimes difficult to teach in your classroom. We also do a lot of work with informal STEM. We have the journal, we have leadership fellows, which are now, we're rebranding those folks as ambassadors, kind of proselytize the SENCER ideals to everybody. We have centers for innovation as well that are set around the country. And we also do a lot of work with K-12 folks too. But the most important thing, and I like to do this is my wizardry, the magic of SENCER is in fact the people, the expertise that we have and the mentoring that we give, right? So I've been mentored exhaustively through SENCER, and I always ask my friends at SENCER what to do. Okay, so that's really important. So the SENCER ideals, just, you know, so if I was to frame SENCER as what it actually is, it's a philosophy. It's not a thing. It's not something you can really measure. It's a philosophy. It's a way of looking at your teaching. And these are the 4 ideals that we live by in SENCER. SENCER connects science through civic engagement by teaching through issues, complex issues, capacious issues, right? Hard issues to solve. We ask the students to put their knowledge to use tackling these issues, right? We also tackle something that we rarely do. We talk about the limits of science as well. So science isn't all powerful. There's loads of problems, right? And then it also helps the students take issues from local to global, right? So you start locally, something that the students care about, and you can relate it then to the bigger picture, the global context, right? So it's nice when you think about it that way. It's a philosophy, a way of doing, all right? So on we go. So I moved to Texas recently, a year and a half ago. And, you know, having moved from New York to Texas, I was like, well, you know, what's going on? And it just so happens that early in March, there was this river authority report saying that the San Antonio River, there was an oil spill in the San Antonio River, right? Now, if any of you know anything about Texas, I live in the Pearl, I live in the Riverwalk, the San Antonio River goes through, and it's a hugely important tourist destination for Texas, one of the most important tourist destinations in the state. And here we have this oil spill in the very precious San Antonio River. But if any day you look at the news, right, what do you hear of in Texas? In particular, you hear about PFAS all the time. These forever chemicals is always in the news, right? So all the students will be like, what is this PFAS thing that they keep talking about, right? But in addition to PFAS, we have microplastics out the wazoo, right? In Texas, microplastics are found here, there, and everywhere. Occasionally, we hear about giardia, and I'm a microbiologist, so I'm always tickled when I hear of giardia because that's bad, okay? But there's all the other stuff, wildfires, there's, you know, one day it's burning, another day it's drowning, we have a drought, floods, and sewage, okay, sewage in particular. So all of these issues are issues that I'm sure are happening where you are at, okay? So that brings me to tip number 1, toxic sites in your neighborhood, right? So when you think about this, I want you to think about it for a second, put this in the chat. Do you know of anything nasty or toxic that is in your neighborhood, right? So I'm sure you have somewhere that you're like, wouldn't be cool to design a project around that, okay? So that brings me back to New York. So I spent almost 12 years teaching in New York, and I was teaching in downtown Brooklyn at a HSI MSI called City Tech. And if you know anything about Brooklyn, you would have heard of these two Superfund sites that went straight through Brooklyn, the Gowanus Canal and Newtown Creek. Superfund sites with amazing history of just horribleness and just terrible. And if you go down to these sites, occasionally you'll see people kayaking, you'll see people fishing, and they don't generally know just the history of these sites and how bad it was. Now, as things are developing down there, we're seeing gentrification, they're starting to beautify the sites, but as you can imagine, people have been living there for years right beside these incredibly toxic waste dumps effectively, right? So we designed a study with my undergrads to look at these sites over time. Now, I know these are a little bit blurry, but I want you to see these. This is Newtown Creek on the left, red sites, the little red circles indicate where we sampled in both of these sites, and we kind of got a bit further out with Gowanus Canal. But we also did surveys of the Greenwood Cemetery, which is this beautiful site, lovely biodiversity, you know, turtles, egrets, parakeets, beautiful place, right beside where these horrible Superfund sites are with the students. And we did microbiome analysis. Now, I know, again, this is blurry. I apologize for the resolution, but any of you who know about microbiomes, we're just looking at amounts, relative abundance, right? So we did it for 2 years, fall, spring, summer, and winter. And we did it for the Superfund sites on the left-hand side, which are classified as brackish sites. And on the right-hand side, we classified the cemetery as freshwater sites. And as you can see here, this is actinobacteria. These are the friendly bacteria that give rise to all the lovely antibiotics and everything else that we want to see. And we see an overabundance of these types of microbes in the cemetery, all right? Whereas if we go to, let me see, oops, my computer's being a bit slow. Come on, computer, you can do it. You can do it. There we go. If we go to proteobacteria, poopy bacteria, you can see that there's an overabundance of poopy bacteria, fecal associate bacteria in the Superfund sites. So they're learning molecular biology, they're doing microbiome analysis, but they're doing it in the context of these Superfund sites that just so happen to be right in their neighborhood. So the students are getting a sense of, wow, this is right under our noses and we didn't know, okay? So if you don't have access to a sequencer, you can do other things though. So Riverkeeper in New York City has community science data that looks at counts of fecal coliforms and enterococci. So you can interrogate citizen science data and bring that into your classroom, right? And you can look at that. And then again, the Hudson River, just like the Superfund sites, Hudson River, 900,000 gallons of sewage is dropping into the Hudson River, all right, every year when it rains. So what we did with the Hudson River was we took Petri dishes, standard Petri dishes, and we added heavy metals to Petri dishes. So cadmium, arsenic, nasty stuff. And we took water from the Hudson, plated it on these heavy metal plates. We didn't have money to sequence at the time. And we found lots and lots of colonies that could grow happily on these heavy metals. So this basically shows that there's microbes in the river that can handle these heavy metals, which serves as an indicator that there are, in fact, heavy metals in the river. So you can think about this, rivers work too. You don't have to have a Superfund site. I'm sure you have a river in your neighborhood that you want to sample from. So SENCER faculty, I'm not alone in this. So SENCER faculty are really good at looking at issues that are local. So the Flint water crisis, we all know about this one. So we have a research project that was designed on the Flint water crisis, and we've others that take geology courses and look at the river and tackle it from a variety of different facets. So from wastewater treatment to drinking water quality and so on and so forth. Entire courses built around these types of issues. So I'm not alone in this. There's loads of resources out there. So second hint, sneak in the science. So I don't know if you've ever done this, but sometimes you're going to be dealing with a class of students that are like, I hate science. Science isn't for me, right? So sometimes we have to be sneaky about it and sneak it in and, you know, through subterfuge, teach them science, even though they don't realize they're learning science as they do it, right? So this is a course I developed. This is one of my favorite things. My father is a gas fitter / plumber. So I've grown up fascinated with toilets. So at the new school, I had the opportunity to teach a toilet change the world class. This was a freshman seminar. And if you've ever taught freshmen have everything to learn when they come to college, right? So they don't know where anything is and so on and so. They don't know what they're doing, frankly. But this course was an awesome opportunity, and I taught it through field trips, projects, and the project was to design a better toilet, right? So we all know toilet design hasn't changed very much in decades, so we need to do this. And these are all high impact practices as would be defined by the AAC&U. But we also brought in social justice, environmental justice, gender equity, and everything else into the course. So here's an example. So if you're thinking about this, look at your campus, right? Look at your campus. There will be something on campus that you can visit. So the new school had a wastewater plant in the basement. Now, many of our facilities have wastewater plants, or they'll have chillers, or they'll have generators, or they'll have something mechanical that you can bring your students to. No one had ever brought the students to the plant that was in the building. And they were very confused when I said, I want to bring my students to the plant. They're like, why? And I was like, because they could learn something. Instead of bringing them off to Newtown Creek, I could just bring them downstairs. And the facilities guys were delighted to do this, by the way, and they showed us everything. And students who came to the new school to be acting majors were asking about reverse osmosis systems and why we would spend the money on these kind of things, and why were we venting the gas? Why weren't we using the gas generated to heat the water, and so on and so forth? Brilliant questions from a bunch of students who didn't want to be science majors. So this was cool. I also use e-portfolios. So if you've ever used an e-portfolio, it's a reflective practice. It can be as simple as a Google Doc. They write on a Google Doc. But this was something where we used our world in data, and we looked at the census data with respect to who had toilets in the states, had the students look at their own state, who has access to a toilet in your state, and they were all shocked to find out that here in America, a lot of people don't have access to showers, toilets, flushing, sanitation. A lot of people in the States are on septic systems. Some of them are not on sanitation systems at all. So students are reflecting on this and getting quite a surprise when they do that. Another thing I do is through SENCER, we have, and I'll say this, we have kick-ass girls who are doing awesome work around the world. And so I have a colleague called Cheryl Broverman, who's in Kenya, who's working with a school that she established in Kenya. And I had her kind of zoom into my class. This pre-COVID, she zoomed in, and my class learned about what was happening in Kenya with respect to educating girls. So you can use experts that are in your network and have them come visit your class via Zoom. You don't have to do a seminar. Bring them in on a Tuesday and just have them educate your class about what's going on in the world. And lastly was this, and this is, I'm very proud of this. So during COVID, when my students were all over the world displaced because we were in lockdown, my students were able to design, I don't know if you can see this very clearly, but this is a cardboard prototype of a new porta potty, right? And if you see here, this is a composting toilet here. So it goes down into a composter. They were using rainwater to wash the hands. It had solar panels. It was all sustainable, and it was culturally sensitive. So folks could actually squat on the toilet. It was designed so you could squat on it should you wish to. And this was kind of the pinnacle of the year. Beautiful thing. All right. So toilets, everybody has to poop. Okay. So the second thing is, if you can get your students to understand that science can be used to make money, they will always engage with it, right? So showing them the money is where it's at, right? So you can make money from science. And the way I do it is through biogas. So in Texas, biogas is not top of the list for sustainable energy, right? So, you know, our electrical company just said they're going to switch from coal to natural gas. And I was like, well, you couldn't get more natural than biogas. So let's do biogas instead, right? But biogas is made during the wastewater process, right? And a lot of utilities around the country are actually starting to do biogas and use it, right? So you can have your students learn about who's doing biogas and where it's happening and how much and so on and so forth. And they can look at their state and they can figure out what's going on. And so my students were tickled to find out that, you know, we were studying Newtown Creek, remember the creek I showed you, and the wastewater plant in Newtown Creek is generating biogas. So they were like, oh, that's kind of good. That's good news. So we're doing something good for the planet, right? And when my students learned about biogas, and I taught them about biogas using a K-12 method, right? These are Coke bottle fermenters, digesters, right? And so it's manure that you buy on Amazon, all right? You can get Amazon to deliver manure these days. This is manure, and they're food of choice. And basically there's an overturned bottle and there's tubing going up to the other Coke bottle. And basically the gas is generated and they measure the gas and they kind of write in whiteboards. And again, these are new school students, not science majors, learning about biogas. And when they came with their project designs, their toilet designs at the end, a lot of them actually integrated biogas into the design. So I saw that as they're beginning to think systematically. And they even came up with ways how we get the biogas around, you know. And Heather will like this. This is a Greg Eckhart quote. It's only wastewater if you don't use it, right? So waste nothing. There's value in poop and you can make money from poop. All right. So I hope you're thinking about this, right? How can you show students that they can make money from science? And I'm sure you have loads of ideas, right? From making cheese to selling honey to coming up with new ways of doing things, right? There's tons of ways you can get connected to economics, right? And that will always make your students excited. The last tip I'm going to talk about is curing your teaching. So what do you think of when you think of curing your teaching? I'm hoping that you're all thinking of classroom undergrad research experiences, right? So classroom-based research experiences. These are probably the best ways you can get your students to think about science, and it's an inclusive method. Every student gets to participate in this. I've been doing this for years. I'm all about ensuring that my students do not have to give up their day jobs to be scientists, right? They shouldn't have to. If they're coming to class, let's get them doing science and learning about it at the same time. Now, if I was to give you one of my favorite cures that's out there, I have my own, as you can see, we were doing stuff with microbes and everything else, but one of my favorite cures that's out there is Tiny Earth. So if you've not heard of Tiny Earth, I want you all to research Tiny Earth tonight for homework, because Tiny Earth is one of the best examples of a community that are devoted to and committed to diversity, equity, inclusion, justice, and allyship, right? And what they've done is they've taken examples of cure learning objectives, which you'll all have seen, right? Distinguish between control and treatment groups. They've talked about how you can adapt this using AGELI principles, right? And then how you can assess and make an activity to address this, right? So they actually give you concrete examples of how you can take a learning objective, transform it so that it's taking into account diversity, equity, inclusion principles, and then how you might actually do it. So they have a beautiful paper in GEMB on just that. And so check out Tiny Earth. They're fabulous, right? It's a great entry point for microbiology classes, all right? And this, I just want to show you this because they did something else which I really liked. So we're all doing these land acknowledgements, right? We're all, I'm going to acknowledge the land. But they're taking it one step further and saying that, soil is alive. It's a being, right? It's a thing. It's something we can go and exploit, right? So when they started looking at soil as being a living thing, and once they did that, for me, I was like, soil is alive. Of course it is. Why wasn't I thinking of it that way? We're exploiting the soil when we take it. So we're not acknowledging the land. We're acknowledging the fact that soil is alive, and we shouldn't exploit it, right? And so I really, really, really like that. So check out Tiny Earth. You'll love it. Now, it would be remiss of me to not mention REMNet. So Research Experiences Microbiomes Network. This is a network that Carlos is in, and he's joined me in this, and Melanie has been involved with this as well. So we're a network that we're trying to get people to do microbiome related pedagogy, okay? And we're trying our best to do this, and we have workshops and we have resources and everything else. And if you're in ASM, we have webinars that are now going to be freely available on YouTube that you can all go in and have a look at. And we're always looking for new things to do. But you can get in touch with me about REMNet too, or Carlos or Melanie. But this is basically REMNet. So I am sure there is a complex microbial environment that you can sample. That could be anything. It can be the air, it can be the water, it can be the soil. And if you're me and you can't get outside, it's going to be the surfaces of your school. Elevator buttons are a personal favorite, hand dryers, and so on and so forth. So you isolate DNA, extract the DNA, and we use primers targeting the 16S ribosomal region of the microbe, and then we sequence it. Now, I'm extremely privileged. I have a sequencer, right? Not everybody does, but REMNet will provide means for you to do this, okay? But we also show you how to analyze it and how to blast it and everything else, right? So we can give you the tools to do all of this, right? Be it everything from pipetting to doing the experiments, or we give you data, right? So you can do it with your students. You won't be excluded, okay? And just bear this in mind. Human microbiomes are hard to do, right? Because of compliance and IBCs and protocols and everything else. But the built environment is a beautiful thing, right? So for me, I'm studying wastewater, but you can do surfaces, you can do houses, you can have the students sample their house if you want. You could have them do commonly touched surfaces. You can consider the airflow of buildings, which is cool, okay? Settle plates, all of which is incredibly important given the pandemic that we just went through, right? But also you can think about the outside, right? From tree pits to window ledges, to green spaces on campus, to fountains outside, they can sample that too. And if you do want to go further with this, you'll just have to consider compliance, right? And so if you're going near animals and so on and so forth, you'd have to have permissions. But there's a world of microbes out there that you can sample. And if you're thinking about experimental design, this is what we came up with during the COVID pandemic, right? Because we were, struggling as to how we could do this and keep it going as everybody was at home or trying to do things under lockdown. But all of this, anything like that's in blue can all be done online, right? And then all of the red stuff here. So we start with our experimental design, coming up with a question, sample collection, DNA quantification, And all of this part, the PCR amplification gel analysis, all of this, everything can be done through simulations, right? So we have, again, we've got these cartoons, we've got videos, we've got virtual labs. All of this can be done through simulations. So you can take each of these blocks, you can spread it out over 2-3 weeks, you can do some of it, you can do all of it, and you can adapt this kind of framework for your micro lab. Works really well. So Tiny Earth is a dream piece of cake for a standard microbiology lab, but this would also work in a microbiology lab. You just have to figure out what you can and can't do at your institution. So there's 2 ways that you can cure your teaching. And remember, if you sensorize it, right? So you sensorize it, you take the issues, right? So I'll give you an example. With Tiny Earth right now, my students are sampling soil for microbes, right, that can produce antibiotics, which is the backbone of Tiny Earth. But we're not just sampling any soil. We're developing across the street from my university. And the developing, sorry, I'm declining a message there on my phone. We're developing a residential area across from my university that was home to coyotes, wild boar. We even had what do you call that? Roadrunners, right? All sorts of cool creatures. And then we're building all these apartments over there, and all the students are horrified. And I was like, well, we can look at this scientifically. Let's sample the soil as they develop it. So we're taking samples from soil that they haven't developed yet. And as they develop over time, we're going to continue to take samples, and we're going to record the impact of this disturbance on the environment. So we'll have scientific data to say, it wasn't just the boar that you got rid of. You reduced the microbial diversity of the soil. And remember, we care about the soil and we want to protect it because it's a living organism that we need to survive, right? So you can do anything. So my mantra for all of you is you don't waste anything. We have no time, faculty. We're all extremely busy, right? I don't want to work this hard anymore, right? So you have to consider, how can you make your teaching work for you as well, right? So when work in the classroom spills out into the lab, right? So this is the spillover you want to happen. And that was a microbiology joke, right? Probably passed y'all by. But you're going to consider how you can combine teaching, your research with your teaching, case reports, projects, classroom undergrad research, and so on and so forth. There's a wealth of things you can do. And the thing is that students that are trained well become marvelous colleagues that will go on to do PhDs and masters and maybe become faculty, or just become your friends for life. That works too, right? But you can have these fabulous colleagues that are your students, right? So you waste nothing. So do you remember I told you I do all this microbiome work? So this is a serendipity moment for y'all, okay? So this is the thing that changed. This is what got me on with This Week in Virology, right? So when COVID happened, I was in New York, and we were, if any of you know what happened in New York, was the worst thing ever, right? So many people died. I can't even talk about it now, right? But in essence, we wanted to do something. We wanted to do something. We felt like we were perilous, right? So colleagues of mine, John Dennehy and Monica Trujillo, got in touch with me and said, listen, we've heard about this wastewater thing they're doing in Europe. Do you think we could do it in New York, right? And we were like, of course we could. John was a virology expert. I was the microbiome person. Monica she had expertise in everything, frankly. So she got in touch with me, and we decided, let's figure out if we can do this. So we knew that we could isolate the virus. We could figure out a way to isolate the virus. But I also had remember, I have the sequencer. So I was like, well, I'm sure we could sequence with my sequencer, right? And what we did was, you ready for this? We took the methods we were using with the microbiomes and just applied it to COVID. I'm not joking. That is exactly what we did, right? So if you think about wastewater, we spent a lot of money during the pandemic swabbing a bunch of people, right? And getting individual data, right? But the thing about wastewater is that you can surveil entire households to neighborhoods to cities with one sample. Just think about that. One single sample, hundreds of thousands of people, right? And it's unbiased. Everybody goes to the toilet, right? Not everybody gets swabbed, but everybody goes to the toilet, right? And it doesn't cost as much because you're only taking one single sample, right? And it's accessible to people because the actual method itself is cheaper. So more people can do this than clinical sequencing, right? And the nice thing about it so far is that the virus isn't infectious. There's plenty of other things that are infectious in the wastewater though. So bear that in mind, okay? But the COVID itself isn't infectious. So we did weekly monitoring of the virus in New York City. 14 wastewater plants, 1.2 billion gallons a day of poopy water. And we were able to identify all the variants at the time, okay, which is pretty cool. How did we do it? This will look familiar to all of you. You've all done PCR, I'm sure, okay, but PCR, We looked at the spike. The spike looked like it was the most variable region. And remember, this is, the first year of the pandemic. We took primers to the spike and we designed two methods. I only have an iSeq, so this is a little fella. It's not a huge capacity sequencer, right? But we had colleagues who had a MySeq. These are the rich scientists or the collaborators in Missouri, right? But with my sequencer, we were able to do 333 bases of the spike. And then with the MySeq, we could get the whole, what we call the receptor binding domain of the virus, right? And so we sequenced this. And with sequencing as it is right now, you can sequence all of the viruses in a sample, right? So it's kind of cool. And now this is confusing, but I'll go slow with you. So this just shows the first couple of months of when we were sequencing, right? So if you look along the left-hand side, these are clinical data. This is the clinical data that was being provided by New York. Remember, they were only sampling around they had about 10% of the positive swabs were being sequenced, right? So just imagine that number, 10% of the positive swabs, whereas we were sequencing everything, right, that was in the wastewater. So we had a huge difference, right? But if you look at the colors, just look at the colors of the charts, it's pretty similar, right, in terms of the mutations that we could detect, right? So it looks pretty similar. But what jumped out at us, though, was that in the wastewater, we had this additional segment. Can you see this dark blue segment, which was these weird variants, right, which we weren't seeing in people at all, right? So we were like, we have these weird cryptic variants. And that's what got us on This Week in Virology. So any of you who are like, how did you get on this? This is what got us on there, okay? And wait for this. So I'm very proud of this, right? But we got into Nature. We got a Nature paper out of it. Like, what are the chances? Now, we had to have a collaborator who had loads of money. But we got into Nature. And this was huge because a lot of the students on here were recent graduates or undergrads and so on and so forth in the lab. And this is CUNY people. I was at the New School. These are CUNY people. We have no money, no resources. So this was a huge thing. So we can make a huge difference, right? We really can. So what can we take from all of this, right? What can we take from all of this? Waste nothing, right? And I do genuinely mean this to all of you. The biggest piece of advice I can give to all of you is that there comes a point where you have to be selfish. Truly, you want to do everything for your students, right? You want to give your heart, mind, body, and soul to your students. Of course you do, but you'll burn out, right? You will burn out. And we've seen it left, right, and center. We're all exhausted, right? So if you can figure out a way to make your teaching become your research, be it scholarship of teaching and learning, be it generating data for little grants, whatever it is. Carlos is an expert in this as well. Everything we do in class, we're generating data, right? Melanie's doing this too. There are many ways you can do this. And the second part of this is if you teach true issues, right, you can reach the SENCER ideals, right? So this is important. So how can you make students realize that science can be a solution, but it's limited, right? It's limited. How can you make an impact on social issues in your area, right? This is important. How can you connect what's happening locally? So if you find microbes that are resistant to heavy metals, that's hugely important. That means there's heavy, like we don't hear about the Hudson River and heavy metals, right? And I'll guarantee you there are heavy metals in every place that you search, right? But we just don't know they're there, okay? You can use the data that already exists. You don't even have to generate the data. You can look at data that's there, that's just sitting, waiting to be analyzed by students, right? And when you do all of that, you realize that STEM education is civic education, right? Because you're getting your students to realize that what they're doing relates to the real world, that they can have an impact, a real measurable impact in whatever shape or fashion it is. I'll give you another example, right? During lockdown, I had my students use glow germ. Do any of you know what I hope you all know what glow germ is, right? It's just this little liquid thing. We couldn't do any experiments, so I sent them home to use glow germ to identify high contact surfaces in their households, right? So where are all the high contact surfaces? Because they have preconceptions of what these are, right? So they designed experiments at home, right? And then we made a manual for facilities, not kidding, to identify high contact surfaces. Now, I don't think they ever used it, but we made a manual so that they could identify high contact surfaces and say to facilities, this is where you should focus your cleaning efforts, because what you're doing right now is wasting money, driving resistance, and so on and so forth. So you can be very creative and come up with ways to do things, right? So SENCER's greatest resource is all of you. Thinking, innovating, sharing, sharing about tools, sharing ideas, right? And then if you're not sure what to do, right? If you don't feel comfortable doing this yourself, Tiny Earth, others, Remnet, you can reach out to them and they will help you design something, right? And so remember, especially Tiny Earth and Remna, they're very open. So if you come in and say, well, I see what you do, but I want to do something different, can you help me do that? 100%, of course you will, right? So, and as any of you, if you reach out to me, I will definitely help you figure out a way to make research and teaching work for you, because that's the way it should be. We're too busy. We're too busy. All right, and that's where I'm going to leave it. I'm going to leave loads of time for questions and discussion, because I know this is a group that needs to do that. So so if you all take your phones and you boop this thingy here, it'll bring you to the slides, right? And if I just show you very quickly, I'm gonna just show you real quick. There's loads of other resources in here. I put extra things, right? So you can find other things, especially if you're into water. So just boop that thing. I hope it works. See, you all have the slides. And I do see cameras going. That's good. That's a good sign. All right.
Davida, can I post these in our Slack as well? Is that good with you?
Of course you can. Absolutely. Absolutely. And once you're all ready to go, I'll stop sharing so I can see your faces. All right. You all ready to go? I'll stop sharing. All right. Now I'm going to look at the chat because I can't do two things at once. All right. Okay. All right. So hit me with questions and tell me what you're thinking and give me your ideas. Well, maybe not hit me with questions. Send them my way. So how many of you had heard of SENCER before? Had any of you heard of SENCER before? A couple of you had. Okay. I see some of you actually. That's great. That's great. So are you thinking, what ideas do you have now? So did any of you get any ideas from what I just said? I hope you did. So stick your ideas in the chat. Let's see what you're thinking, or talk to us right now. Turn on your mics there and talk away. Let's have a discussion. You're all very shy.
Dr. Smith, I have a question comment that really clicked with me.
Heather, you can call me Davida.
That feels weird. That feels weird. Davida. So I totally connect with STEM education is civic education. But then I realized STEM education is civic education is workforce development. It just really hit me. And I just love how you do that and how you bring it all in. And then I think, it puts your students in touch with, all the jobs that, in your case, biologists can do and stuff, including for the city and municipal, utilities and just everything. And so that really kind of connected for me today. So thank you so much for that.
Thank you, Heather. And in fact, because I wanted to leave enough time for us all to have a discussion, I actually had slides on workforce development that I removed because I thought, it might be too much and I want to leave time for y'all. But wastewater epidemiology, that last part I spoke about, that is an emerging, area for workforce development. And Heather and I are actually working on various strategies to try and show students that water workforce careers are cool and exciting and you should be considering them. A lot of the biologists don't know that there are careers to be had in the water workforce. The water workforce is aging. It's not as diverse as we would like it to be. There are huge opportunities for growth in that sector, and you can do amazing science there, right? So some examples right now, you can monitor community associated infections. So for example, have any of you heard of Candida auris, for example? Who's heard of Candida auris? I hope several of you have, right? So Candida auris is an emerging, incredibly resistant fungus, which is terrifying. If you're all watching The Last of Us on HBO, Candida auris is the story of, that I'm most worried about. Nevada, the city of Nevada, a paper just came out where they studied an outbreak of Candida auris using wastewater. So this is a job opportunity, and I'm trying to get, like Heather knows this, writing grants left, right, and center so I can start wastewater training for students at my institution. Not being as successful as I'd like, but I'm trying, okay? But the rest of you, it's hard to work with wastewater, right? It is, but you can develop tools that can be used for wastewater, right? So think about it. Remember I mentioned Giardia? Develop an assay for Giardia and then connect with someone who's doing wastewater analysis and they will try your tool, right? So think of it that way. You can develop the assay in your lab, which is BSL1 or whatever, working with DNA, and then the scientist who's got access to wastewater can do the wastewater work, right? So consider that. Any other thoughts or questions? Don't be shy.
Davida, my question is always going to be around faculty development, because that's kind of my passion area. And I've worked with faculty who love SENCER and the concepts, and I've worked with faculty who are like, that's not how we teach science.
Yeah.
So I always wonder, how do I, as a faculty developer, how do I reach people who may be recalcitrant to try to, you know, engage students civically in their STEM classes, because they're STEM, you know? So
Yeah, I do. Did I hear somebody else say something there? I don't want to cut you off. If somebody was going to answer that. No, truly, to be honest with you, I've met many of those faculty too, Heather, over the years, right? It's not, and a lot of the time, just like our students, there are factors at play as to why faculty think that way, right? It's not like they're just like, ew, civics. It's not. Personally, I always say this to faculty, foreign faculty in particular, right? Folks who aren't from the States, a lot of us are on visas, right? And we're a little bit wary of getting into civic issues, right? But workshops where we actually come and do, I'm not joking here, workshops where we actually come and we offer a workshop, that's usually how we get people to come around. Because what we do with the workshops is that we invite folks from all different aspects of the school. We invite the humanities people, the faculty development people, the Civic Engagement Center people, you know, the student success people, deans, you know, all of those folks to come. And we deliberately try and put everybody into interdisciplinary teams. So breaking the chemists out from just being with the chemists, that's probably the most important part. Because when the humanities folks say, geez, I have a great book on, you know, such and such, and wouldn't it be great if we could read some chapters in your class, blah, 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 or would you come to my class and talk about this in my class? I think that's the moment when most people kind of go, there's something here, right? And then the second part of it is making folk realize that civic engagement can be as simple as, are you recycling on campus, right? So civic engagement doesn't have to be writing to your governor, which is terrifying. I wouldn't have felt comfortable doing anything like that. But going into the cafeteria and looking at how many, how we're washing, are we using plates or are we using disposables, right? Single-use cups, and many people are using tote bags, you know, all of that kind of stuff. Where's the recycling going on campus? Do we have a bottle repository on campus? It can be anything, right? Civic engagement can be anything that the students are engaged, taking the science that they have and putting it to use in whatever environment they're in, okay? So think of it that way. But genuinely, Heather, it's usually, one-on-one, it's hard to do. You can find examples, you know, and show them, right? But I think a workshop environment is the way to do, because then they feel like they're part of something bigger and they're like, oh, but having the disciplinary folks together is really, really hard to break into. You have to have someone from the discipline to proselytize. And I found it, I've particularly found it difficult with chemistry, because chemistry has a very prescribed curriculum, where they're like, there's no time for this, but you can break them down. It just takes time. Brittany, I see your hand up there.
I think one of the problems is that with the time, but also there's such a short amount of window in the curriculum. For me, I'm a physics instructor, so like physics 2, I talk about the alternating current and direct current wars and all the deaths that happened in that process, and the business transactions. But in my physics 1 component, trying to figure out how to bring civic engagement in my kinematics topics, but then also cover the topics. There's so many topics to cover that you're limited to. And so I think that's one of those big problems. And when I go to these sites like SENCER, there's very few
Physics models.
Physics stuff on there.
I know.
And it's,
well, you ready for this? You ready for this? So we actually have some folks who are working on physics stuff that I'm on a grant with, and I'll actually help you with that later. It's not going to be that. It's more NMR and spectroscopy and that kind of stuff. But you ready for this? Simple thing. Every day, when students are filtering into class and they're getting to their seats, and you're trying to kind of, you haven't taken roll yet. Instead, find something that happened in physics that's cool, in the news, in popular media, whatever it is, and just talk about that for the first 3 minutes of class, right? That's probably the easiest thing you can do. So look at what happened in physics now. And you could even find somebody on social media that did something funny or a meme or anything like that, right? But just start them thinking about that. And then at the end of class, consider a 1-minute paper on what we learned about today. How might you relate that to the real world? Or could you imagine this being of value to your real world or whatever? And if they all come back to you with a no, the next week's class, you start the class with why it was important, right? So that's really easy to do. And so I get it. Biology 1, the first biology class, tons of content, right, that we have to get through. But you can, remember I told you this, you can kind of sneak it in. So using, for example, examples of, assign them a physicist that made a huge impact in the world and have them do that as a project, right? For extra credit even, right? There are ways you can do it. There are ways you can do it, but it's harder. And like I haven't got as much expertise in physics, but we are recruiting folks from physics. So I've spent more time with the chemists. Hope that helps. It's at least a start. I'll put my thinking cap on. Janie, I see your hand up as well.
Yeah, I was wondering if there is are there subgroups within SENCER that focus on certain type of topics? Like, for example, mine is really human medical genetics. And I was also wondering whether there are people that work on the curricular component. Like, are there people trying to develop these civic engagement projects that also interweave it into courses that we can all try out at our different universities and start collecting data on it, and also an emphasis on interdisciplinary integration of the curriculum so that students know that it's not just this one discipline, it's really, an interdisciplinary learning process.
Well, if I didn't emphasize that enough, that is the essence of SENCER. So unlike other disciplinary organizations, most of our folks aren't even scientists. So the executive director of SENCER is, are you ready for this, an art historian. We have examples of courses, Janie, such as stem cells and social justice, right? So we have actual courses, model courses that are exactly that, taking like the story of HeLa cells, we all know this one, but bringing in poetry and art and other elements of the humanities into the class as well. So, and at the new school where I taught, for example, science and politics of infectious disease. So it was science, but it was sustainable development, neglected tropical diseases, and how that impacts local economies and so on and so forth. So there are loads of examples of this, but what we've done with SENCER recently is we've restructured, and what we're doing right now is we're starting to build upon the model of pulse. So I know that Jackie is here. So we're thinking about developing circles. So we're starting to kind of not quite disciplinary circles, but topical circles. So for example, we have a chemistry circle that's working on toxics in building products, right? And I'm going to be working on water, folks who want to do research on water as a water circle, right? So we have a ton of folks who do medical stuff, right? So really pretty exhaustive resources actually on medical genetics and cancer in particular. So if you're all interested and you want to start a circle or be involved in a circle, let me know and I'll sign you all up. Yeah, it's just like we just started that restructuring in the fall, and we're going to start in earnest this year. But if any of you are interested too, as being what we've been recruiting to are ambassadors, right? We have an ambassadors program where we're trying to recruit people who will be SENCER advocates at their campus. And what we want to do is basically me or you or whomever, we can bring some SENCER folks to your institution and have a faculty development workshop. That would be the ideal. And what we would do is we'd help you organize, we'd help you recruit, we'd help you build it, we'd help you with your curriculum, and we can do it for you. Now, we don't have a lot of money, so it could be half a day or something. But if we can work with your faculty development people, we'd offer a workshop, and our expertise is in working across the disciplines. So my last 3 papers are with an English professor. So just to give you an example, so and we do work really well with other disciplines. I hope that helps. Cool. Any other comments or ideas, thoughts? But yeah, I mean, if you're thinking about it, so we will be having, just so you all know, we have regional meetings, and the regions are kind of getting up to scratch right now at the moment to think about what they're going to do. So we don't have regions necessarily beside all of you, but we have regions around the country that you might be able to get to. But the Summer Institute is going to be the first weekend in August. So get this on your calendar. We haven't set the date yet. It'll be on Zoom, and it's going to be about really strongly about how we get elements of our action to save our democracy, for want of a better word. What scientists can do to save our democracy. And that's going to be kind of the theme, right? And bear in mind that my context is, I don't want faculty to be afraid of losing their jobs, right? So how can we advocate for faculty to help their students be advocates without feeling fearful of doing so, right? So, and trying to build a movement to kind of try and protect this. And we've colleagues from who are in Florida in particular that are going to come in and talk about what's going on in Florida. So if you're all interested, I'd love to see us there. It'll be on Zoom. Lots of examples of people who are doing what I do, and better examples, frankly. And you'll learn a lot of what's going on with SENCER. So I'd love to see you all there. All right. Any final thoughts? I'm going to try and copy the chat so I don't lose any ideas from you. Any final questions or thoughts? I know it's Friday and you're all tired. All right. I think people are beginning to filter out. I'll hover for a while and see if anyone wants to chat.